we'll make a start. So what we're going to be doing logistically today, we're actually going to split into two groups, one group in here and one group in the theatre, which is just next door. And I've forgotten which one is which, but I guess the one that Peter's talking in first is in here, and the other one is next door. Also, if you need transportation to the airport or somewhere else, then there's some sign-up sheets at the registration desk. So what I'm just going to spend a couple of minutes doing is sort of describing what it is we're going to do the rest of the day. So we're going to have, for the first part of the day, 2 parallel sessions with the same kind of TED-like talks and panel discussions that we had yesterday, which was generally well received, I think. I was probing a lot of people. And then in the afternoon, we're actually going to spend time beginning the process of writing some kind of proposal for what remains to be seen. Could I get the first slide? So what I did yesterday evening and this morning is I went over all of the output from the sessions that were held yesterday afternoon. And I tried to effectively summarize those around, and we're going to go into this a bit this afternoon because it's going to define what we do this afternoon. But, okay, here it comes. And I did it. It will be there shortly, I'm sure. No, that's not it. Well, I can just talk around it. So I essentially broke things into 4 areas, and one I would say is the challenge and the scope of what any enterprise would be if we decide to proceed with such an enterprise. The second was around infrastructure for supporting whatever the enterprise would be with respect to its scope. The third was really around dissemination and education and effectively usage. And the final one was really about sociology. So it was about reward systems. Okay, so this was the challenge part of it, and I think that I don't want to go into all of this now. We'll do this afternoon, but I think as we go through this morning, the key things to me, at least, and certainly if you have thoughts about this before we break, it'd be good to hear them. But I think we need to really define the biological problem and the scope of what that is that we would attempt to put into this in say, the next 5 years. And without that, I think everything becomes too nebulous. And I'll be quite upfront with my own thinking about this, is that based on projects that I've seen, is so, for example, I think the Human Genome Project had a very distinct goal, and lots came from that at different time points, and it took much longer than people expected, but it was something that people could generally grasp hold of as to what the implications were at the end of that time frame. If I look at some other large projects that I'm familiar with, say, for example, the structural genomics projects, it's not that they haven't been valuable, but what the goals were has been a moving target over time. And I don't think it's captured quite the imagination and the community, broadly speaking, that one would like to have around such an initiative. And so the idea that we have something that we could all take away and grasp as a deliverable, so whether it be, I'm not going to put words into people's mouths, this is what we'll have a brief discussion about, but it could be we take a very simple cell type and we agree that at some level we're going to describe, have fully available within some resource the effective structure of that cell type and some simulation that relates to specific biological processes around that given cell type. So that would be one possibility. But that would seem to me to be important. And as this is a collective effort, do people have comments about this at this point? I don't necessarily want to go into all of these different sort of findings that came out of this. Terry, I think we should use the mic if we've got it. Oh, here, let me bring it to you.
So the NIH funded such a project, I think about 10 years ago. Al Gilman put together a large team. I think it was like $25 million.
The cell signaling initiative? Yeah.
I think they picked 2 cells, and they tried to collect as much data as they could about those 2 cells. And I'm not sure whatever came of it, but I don't think they ever got to the point where they could do any modeling. But, you know, it's been tried, I mean, to focus on 1 cell.
So with regard to that, I mean, not much, they got a lot of data, but it was really a premature thing. So they actually did have a lot of data from the cell signaling thing that's just sitting there as far as I know. But my question to you is whether, since this is NSF now, would we be precluded from doing anything that would have relevance for disease, like how you have a normal system and how that's then perturbed by disease? And that seems to me something that could be a really good way to look at any model system, but really then explore that aspect of it. Would that be excluded because it's NSF?
I mean, I would look at it not that way. I would look at it from the point of view, let's define what it is we want to do, irrespective of what the respective funding agencies think about. And then, you know, I think this, you know, seriously, this would be big enough that would potentially transcend agencies. Or it could, it could be a not, you know, it could be if we came together on something, it could, you know, it doesn't have to be a solicited entering a solicitation. We could actually put in an unsolicited application wherever we wanted as a group.
Right.
And that would actually be a fun thing to do.
Right.
Mark?
So in moving from a whole cell in its tissue environment to the molecular constituents, we already know pretty well where the hardest gaps to fill are and how to fill them, but it takes time and effort. Layering on top of such a completely described cell at all scales, identifying all of the key molecular constituents, requires definition, or at least a clear articulation of what models from that need to be extracted in order to serve certain types of simulations. So I think that's part of the work that we need to do in groups, is to define for different ways of thinking about the dynamics that we can't actually measure currently with new physiological, optical methods, whatever, FRET, the like. What is it that we need to continue to characterize or sketch in to identified cell problems? And then how do we spin those into the right simulation environments so that one can ask questions that you can't ask by experiment today? That's a grand challenge, at least the way I see this part of the field.
I would agree. I mean, I don't want to, we're going to dwell on this more this afternoon. I just want it to be buzzing around a bit in your head as we go and hear a whole bunch of other incredible science that you're going to hear today. Do other people want to make comments before we break for that? Sorry, I keep forgetting to say your name, because just for the people who are remote.
That's true. Herbert Sauer from University of Washington. I want to comment on what you said. I think it's the wrong way around. I don't think you want to start with filling in the gaps and then building the model. I mean, we said this yesterday. You start with a model, then you know what gaps to fill in. And I think that was partly the problem with the Signaling Alliance project. They wanted to, I was at the initial meeting where they launched it, and the emphasis was on collecting the data. Then right at the end, they said, we're going to build a model. Well, that was completely the wrong way around. You start with a model. It doesn't matter how rough it is, how bad the model is. That model will tell you what you need to know, what you don't want to know, depending on what questions you're trying to ask. So I don't think you should try to fill in the gaps first. You build a model first, then you know what gaps to fill in. If you do it that way, you'll just end up with the Signaling Alliance effort again.
Okay, it appears we're going to have quite a lot to discuss later on. So let's not dwell on, I won't go into even the other points now. Let's get on with the science presentation part.